Actually, when I started neuroscience, I thought I'll never talk about consciousness because it's ill-defined. And I also thought I'll never talk about attention because it's ill-defined. Okay, so about 20 years ago, I realized it's unavoidable. I have to talk about attention because otherwise I cannot relate to the huge literature that is there about attention. And now I am also talking about consciousness. So there's always a good reason to change your mind. So the topic is conscious perception and how to restore it in blindness. So in the first part of my talk, I'll focus on the neural correlates of conscious awareness. And thereafter, I'll be talking about how we apply this knowledge to create a visual cortical interface for blind people. Now I'm going to illustrate some of the concepts that I would like to introduce with a few experiments that I invite you to participate in. And the first experiment is very simple. I just would like to invite you to remember this number. It will come back later. Okay? Put it into your memory. And now I'm going to do a second experiment that is a perceptual experiment. Very simple. I'm going to present a stimulus, a circle about here. And if you see it, please raise your hand. If you don't raise your hand. So 1, 2, 3. Okay, that was easy. I'm going to make it more complicated now. So this is for the very careful viewers. 1, 2, 3. Nobody saw it? Okay. Give it another try. 1, 2, 3. Nobody saw it? Well, I don't control the quality of this monitor. But I can tell you there was a stimulus on the first one and there was a stimulus on the third one on my computer, maybe not here. The second one, there was no stimulus. Nobody committed a false alarm. That sometimes happens, but that's nothing to be ashamed of. It happens all the time. And the point is that for each and every one of you, there will be a contrast level where you sometimes see the stimulus and sometimes you don't. And that's a well-known trick if you want to study visual awareness, because it allows you to present the very same stimulus over and over again, and sometimes it reaches consciousness and sometimes it does not. So that allows you to kind of get insight in what makes a stimulus conscious. And that is what we set out to do a couple of years ago. And when we started to do so, we actually did not expect this, but we got pretty strong evidence that supports the global neuronal workspace theory. So I'll briefly explain to you what this theory is about, although Stan probably already did a much better job than I will. So this is a stimulus that enters into the eye, into the LGN, then is propagated in a feed-forward manner to higher areas, including frontal cortex. And then there is this mysterious ignition process, maybe it's not so mysterious after all, that then accounts for a self-stabilized pattern of activity, probably based on recurrent connections within cortex, but probably also with subcortical structures, that then causes a self-stabilized activity pattern that corresponds to a working memory. Okay, so even if the stimulus is no longer there, there's a trace of that stimulus allowing the subject to report the stimulus that it was there at a later point in time. So this is the experiment that we carried out in monkeys. It was basically the same as you have been participating in just a moment ago. So the monkey on 50% of the trials saw a dot. The other 50% of trials there was no dot. Then there was a delay. And then when the animal saw it, he made an eye movement towards the remembered location of the dot. And there was always this reject dot, allowing the monkey to report that he didn't see the dot. Okay, so a very simple experiment. And of course you can then vary the contrast of the stimulus here along the x-axis, and if you have the high contrast, the animal will basically always see it. Then if you reduce the contrast, perception becomes less reliable, and you can, and that is why we did define two thresholds, one at 80% and one at 40%, and when accuracy was lower than 40%, we said this is a really difficult stimulus. If it was above 80%, we said this is an easy stimulus, and if accuracy was in between, we said this is a stimulus of intermediate difficulty. We recorded from area V1, area V4, which is a higher level visual area, and an area in the frontal cortex, dorsolateral prefrontal cortex, which is a brain region involved in the planning of eye movements, and that's also a caveat. We're not going to dissociate perceptual awareness from the plan to make an eye movement, okay? That's something to keep in mind. You might want to do that in later experiments. We did not do that here. And then we started to record, and we were immediately in for a big surprise. So when we recorded from primary visual cortex, here there are the three types of stimuli: difficult, intermediate, easy. Of course, activity increases if you make the contrast higher. No surprise there. But here we sorted the trials in which the animal saw the stimulus in green and which he failed to see it in red. And here we matched the contrasts. Okay, so if you present the stimulus and if it elicits a strong response in V1, it has a higher probability of being perceived than if the very same stimulus elicits weaker activity in V1, as if there is some variability in the propagation of information between the eye and the primary visual cortex. I was not expecting this. Then there's something else that is interesting. So you see that the same effect also holds up for the easier stimuli. Here the effect is still there, although proportionally it's smaller. And you may notice that the easy miss stimuli elicit actually even more activity than the difficult seen stimuli. So reaching a certain level of activity in primary visual cortex is no guarantee that the stimulus will be perceived, as if information can get lost downstream from primary visual cortex. We did the same thing in area V4, and we basically got the same result. Also here you see that activity elicited by the the easy stimuli that are missed is stronger than that, or at least as strong as the easy stimuli that are seen. So also here, apparently information can get lost downstream from area V4. Now the activity was categorically different in dorsolateral prefrontal cortex. So here, whenever the animal was going to report the stimulus, we get this high activity level, and when he failed to perceive it, we got this low activity level. You see that if the stimulus was easy, activity still ramped up a little bit, but apparently not enough, as if there is sort of a threshold that you need to reach for being able to report the stimulus. Now we also looked at the activity on those trials in which the animal failed, in which there was no stimulus, and either the animal correctly reported that he didn't see a stimulus, so that's correct rejections in black, so then the activity is always low. And there were these trials in which he reported the stimulus that was not there. It's called the false alarm in blue, and you see that in V1 and V4 there's actually a little bit more activity than on the correct rejections. So here the animal is sort of hallucinating a stimulus. And you see that in dorsolateral prefrontal cortex activity reaches the same level as the hit. And it looks like a slow ramp, but we have a suspicion that what's going on here, that there are spontaneous ignitions that are actually causing a fast ramp. So from your view, it goes rapidly upward. But if these events happen at different time points in different trials, if you then do the averaging, it looks like a slow ramp. We cannot prove that, but that's just our suspicion. And so if you think about a signal detection theory, so this is the distribution of activity according to the signal detection theory if a stimulus is there. So these would be the hits, this would be the misses, and this would be the distribution of activity without the stimulus. This would be the correct rejections, and this is the false alarm. Then, well, if there's one brain structure that complies with the prediction of such a signal detection theory, then it would be dorsolateral prefrontal cortex and not V1 and V4. Okay, so as if really this ignition process can be monitored in this brain region. And I talked to Stan about that, Stan Hein, maybe 5 years ago or 6 years ago. And he said, okay, I can model that. Actually, I did model that. And this is one of the studies in which this was modeled. And then he said, you know what I'm going to do? I'm going to make the minimum model. And it's a bit embarrassing, but this is the minimal model. It has 5 neurons. So 5 neurons are going to be a model for the entire brain. And it starts with a neuron in LGN, feed-forward connection to V1, V4, parietal cortex and frontal cortex. So activity propagates through these feed-forward connections. There are also these self-connections, and there are also feedback connections. And these so-called recurrent connections are particularly strong between parietal and frontal cortex. So if activity reaches a certain level, then the excitation in those two nodes is so strong that it can self-sustain. Okay, so that's sort of a bistable activity pattern. And if that happens, then you can take the stimulus away, but the activity will stay there. And that, of course, then would be a hit. Now, because of the stochasticity of all these units, if you present the very same stimulus, sometimes it happens that this minimum level of activity for self-sustained persistent activity is not reached. And then if you take the stimulus away, activity collapses. That would be a miss. And you know, this is then the predictions from the model, or maybe these are the post-predictions. So V1, basically you get the same effect, also that you have easy miss trials that have a stronger activity than the difficult scene trials, as we saw in the first part. Before the same, and in frontal cortex now you have this bistable activity. Activity either reaches the high phase or it stays low. And here we know that on those false alarm trials there are sudden ignitions. So what happens, you have stochasticity, and sometimes the activity is just so high that it kind of becomes self-sustained, and then it clamps to the high level. And that happens at different trials at different time points. Okay, so these are the results of the model, and these are the results of our data. And so this was Stan's rainy Sunday afternoon. This was 4 years of hard work in the lab. So here you see the asymmetry between experimentalists and theoreticians. It's just unfair. And of course, this is a very nice support of this global neuronal workspace theory, where we saw basically that if you present a stimulus that is weak but is not going to be perceived, apparently there's not such an efficient propagation of the activity to the higher levels where this self-sustained activity can occur. And if the very same stimulus does reach consciousness, it's propagated more efficiently and then reaches ignition. Okay, so these results, and Melanie may not like it, but this seems to support, of course, an involvement of the frontal cortex, although I do not exclude that it's also the posterior hot zone, right? So it could also be parietal cortex that does the ignition. But this is not the whole story. So we got later results that tell a slightly different and slightly more subtle story. So this is another experiment I would like you to engage in. I'm going to briefly present a complex stimulus and just try to see what is there. Okay, so 1, 2, 3. Did you see something? What did you see? Did you see maybe some texture, brownish, whitish stuff? Did some people see something more? A bird, yeah. So this is the stimulus. So it's a bird. And to see the bird, you actually have to segregate all its image elements from the background. And that requires real work by the visual system. And it turns out that in those cases, the so-called global neuronal workspace needs to reach back all the way to the visual cortex, even all the way to the primary visual cortex. So what happens here is that psychologists call it object-based attention. You basically direct your attention to all those image elements that need to be grouped in perception. We know in the visual cortex this is associated with labeling of all those image elements with enhanced neural activity. And now I want to relate this to consciousness theories. I would like to argue that the item that you're attending is in what people call access awareness. So this is what the global neuronal workspace is about. And the other items, like this stick, that you could attend to if you wanted to, maybe you didn't so far, that would then be in what people call phenomenal awareness. So maybe that is what IIT is about. I don't know, I'm not so deep in those theories, but of course people are frustrated by the global neuronal workspace theory if it claims that the only thing you perceive is what you attend. Because you can attend other stuff if you want to, right? So, and being able to shift attention from A to B implies that B is also there in your perception. Okay, so if you want to, you can direct your attention. Of course, then the two things switch. Now, this is another version of the bird, simpler and easy to use in a lab. So this is a figure ground stimulus. I guess everybody sees a square composed of line elements of a particular orientation that is superimposed on the background of line elements with another orientation. A good friend and colleague of mine, Victor Lamme, studied this many years ago. So this is a paper from '95. And back then he already demonstrated that if a neuron in primary visual cortex has its receptive field on image elements that belong to a figure, then you get a peak response, and then you get the sustained firing phase where the activity is stronger than if the same image elements in the receptive fields are part of a homogeneous texture. So it's a figure, and here's sort of a background. You see, get first a feed-forward response that is driven by the information in the receptive field, which is identical. And then you get this late feedback phase, where the information comes back maybe from frontal cortex, parietal cortex, high visual areas, boosting the activity for figures. Right? So this could be this phase in which you really need primary visual cortex being part of the global neuronal workspace. Now, in accordance with this view, this is another somewhat older paper from Victor's group. So he made the texture, the figure, difficult to perceive. And he's comparing here the trials in which the animal did see the figure. So that is shown as seen. Now note that there's a peak response and then there's this figure ground modulation that's particularly strong if the animal sees it, but it's completely absent if the animal fails to see it. That's quite interesting, right? Because you see that the peak response, unlike in what I showed before, is now there. It's just this late process that fails. So the animal probably saw the texture element. He just failed to superimpose the figure ground organization onto the figure. Okay, so this could be then due to this recurrent processing phase. Now the good news is we see this across species. So this is what we see in monkeys. We recently also, or recently, 2016 we published it, we were able to record from spiking activity in area V3, a high visual area in the human. And there you see the effect. And for our purposes, it's also good to know that we see the effect in the mouse. Okay, so figures elicit also in the mouse more activity in V1 than backgrounds. And that's very important because now we can use these many powerful tools that are now available to study and manipulate activity in the cortex of the mouse. Which is what we did. So this is the work of Lisa Kirchberger. And we used a very strong optogenetic silencer called GT-ACR2. So if this is the figure ground response, you can basically knock out all the spikes from primary visual cortex. See what happens. Or you can just let through the feed-forward response and knock out the late spikes. Or also let through increasing number of these late spikes that are fired during this figure ground modulation. Okay? Now I'm summarizing the data here. So actually we're comparing here mice doing a figure ground task in which they lick to the side in which the figure is displayed. It takes a bit of time, but you can train mice to do this. And there's a version in which they just see a contrast stimulus, so no need for figure ground segregation. Now if you then knock out all the activity, the mouse is largely blind, as you would expect. If you let through only the feed-forward response, but you knock out the late spikes in V1, the animal can do the contrast task at ceiling, but he cannot do the figure ground task. It's not possible. He's at chance level. You really need to let through a little bit of these late spikes where the animal establishes figure ground modulation, and then the animal quickly recovers his ability to perform this task, indicating that these late spikes are necessary not for contrast detection, but for figure ground modulation. Okay, now another thing to note is that if you have a figure ground stimulus and you're directing your attention elsewhere, then this figure ground modulation is much less pronounced. So it really depends also on whether you're interested in the figure or not. Okay, which is kind of supporting this idea that the thing that you're really caring about, that would be an axis awareness, there the global neuronal workspace would reach down all the way back to primary visual cortex, and the other items, they are not as broadcasted as efficiently, right? So they're just left there. And so this is then a scheme that I would like to propose. I don't think I'm the first to propose it. Maybe I relabel some of these elements, because I have a very strong opinion on the relationship between attention and working memory and these different forms of consciousness. And I'm pretty sure that this is going to be very controversial, because there have been many studies looking at the relation between attention and working memory and different forms of consciousness. I think this is probably 90% correct. So forgive me for the other 10%, and it would be interesting to talk more about that. Okay, so the idea is that the thing that you're attending that is an access awareness, the things that you're not attending but that you could attend to if you wanted to are in phenomenal awareness. And of course, that is maybe the most controversial because some people call the non-attended items pre-conscious or completely outside consciousness. So that is definitely worth a debate. And of course, it can be completely outside awareness. So that's kind of also indicated in this Venn diagram. So this is then for perception, and now I would like to make a similar claim for working memory. So at the very beginning of my talk, I asked you to remember a number. Do you remember it? Yeah. So what you're doing is, it's probably lingering somewhere in your brains, maybe in the synapses. Maybe there's still some persistent activity. And then you call it back from memory, and then it becomes an active part of your memory. So then it's brought back into working memory. Okay, I would also like to include that in the definition of access awareness. So maybe it was not there, the number 127, but it is brought back and intended within working memory when I asked you to do so. And then there are forms of memory that's a little bit like the non-attended stuff in the visual world. You could access it if you wanted to, but sometimes you don't. And one example is iconic memory, so I'd like to say a few words about that. And another example is what I like to call accessory memory, and I also like to say a few words about that. And it, of course, may also happen that you lose something from memory, and then it's, of course, not in your awareness at all. Now, a very famous study on one of these other forms of memory that one might want to include in phenomenal awareness is iconic memory. And so I would like to illustrate this famous experiment of Sperling. I think most of you know it. So what I'm going to do is, in this rectangle, I'm going to present 12 letters and digits and try to remember as many as you can. Okay? 1, 2, 3. Okay. So what Sperling found, I can do it again. People are able to remember 3, perhaps 4. Maybe not the first time you see it, but if you get into it, then you'll be able to do 3 or 4. And, well, that in itself is interesting. It probably illustrates the limited capacity of our working memory. But he did also a very interesting manipulation. He did the same thing, but he instructed subjects to only report the items in one of the rows cued by a tone. This tone could be high, intermediate, or low of pitch. And if the tone would be, say, intermediate, then the subjects knew that they were cued to only report the items in the middle row. If it was a high tone, they would report the items in the upper row, and if it was a low tone, they reported the items in the lower row. And what he then found is that subjects were basically always reporting all the letters. That's a surprise, because the tone came after the stimulus was gone. As if there's sort of a fading image of what you just saw, and you can grab it, put it in your working memory if the cue comes early enough. Okay, and that's what is then later called iconic memory. Now, I'm not going to show you the details of the experiment, but we recently reproduced an iconic memory experiment in monkeys. And we basically demonstrated that if you present a brief stimulus in primary visual cortex, we know that, then you get a peak response after a delay, and then a fading response that gradually goes down over about 100 to 200 milliseconds. And it turns out that the number of spikes that are fired during this kind of downslope, that they predict iconic memory. You can mask it, and then you wipe out iconic memory. Okay, so this is one of the types of activity that I would like to include into the definition of phenomenal awareness. I think many people also suggested that that should be done. And this just shows that if you have a mask and you do it at different time points, then the accuracy of the monkey, so that is the behavioral measure of iconic memory, is well predicted by the lingering V1 activity. Okay? So here we have iconic memory. Now a few slides about accessory memories, another type of memory that I would like to include into phenomenal awareness. And now we're going to do human recordings, and this is an experiment that was also set up together with Stan. So what we're doing here is we have patients in the hospital. They have electrodes in the medial temporal lobe, and they looking at a laptop in which we present a sentence, actually 2 sentences, 1 word or 1 name at a time. Okay, so the subject sees the sentence, Barack Obama and Jennifer Aniston sat in a bar. And then the interesting point is there's a pronoun, either he or she, ordered the beer. Okay? Now we record from these cells in the medial temporal lobe that responds to particular individuals. So this could be a cell that is tuned to Barack Obama. So if you present Barack Obama, the cell responds, and you see activity then goes down again. Because then maybe Anne and Jennifer Aniston will be presented. And then there is the interesting condition of the second sentence that starts with the pronoun. And when we present the pronoun he, that refers to Barack Obama, which is the preferred concept of the cell, you see sort of a reignition of the concept. Okay, so maybe Barack was in phenomenal awareness, it was in sort of accessory memory, but now the word he comes, and it is reignited. It becomes part of your working memory, part of the global neuronal workspace. I think that's very important, because if you learn something about the he, that he ordered a beer, you know that the beer actually is now in the hand of Barack and not Jennifer, right? So this is very important for your language comprehension. This is work, by the way, done by Doris Dijksterhuis in the lab. So this is one example of an accessory memory. I'll give you just one more example. So here we present 2 items. Here Barack Obama again and Jennifer Aniston again. And the subject knows that he has to do 2 tasks. The first is to do a visual search, so he has to say whether Barack is there. And then there is another cue, and then he has to kind of recall Jennifer and say whether there's a match. And of course, there would be not a match here. And this is the interesting time point, because now the subject probably is preparing for the search, so Barack should be kind of in a working memory. But Jennifer should be remembered for later. Okay, so that could be an accessory memory. And what we find is that the search template actually elicits strong activity, but Jennifer is also slightly there. She's not completely gone. So it's just these accessory memories that they are still there and maintained by some persistent level of activity that is lower than the active working memory. Now if Barack is not at all in the display, then activity is even lower. Okay? So this is another example of an accessory memory. And so this is now with a picture of the global neuronal workspace, and basically I don't need to repeat what I suggested before. Okay, I look at Walter. I think I have 3 minutes left.
For the talk, I have 10 minutes for discussion. You know?
Okay, yeah, should be fine. So I'll briefly then go over the visual prosthesis work. So a number of years ago, we realized that we are putting increasing numbers of electrodes in the visual cortex. So standard in the lab was about 200. And then we thought, well, why don't we increase this by a factor? So we went to 1,000, because that gives us the opportunity to directly stimulate the visual cortex, which would be very useful if you're blind, and if you have seen in your development to kind of restore a rudimentary form of vision. So in the world, there are 40 million people who are blind, and many of them will not benefit from a retinal chip, because in many of the people, the connection between the eye and the brain is completely lost. So we're interfacing now with primary visual cortex. We're putting Utah electrode arrays. The logic is the following. If you stimulate one electrode in visual cortex, which is a map of visual space, the subject will see a dot of light at the corresponding location in the outside world. If you have 1,000 electrodes, you can create 1,000 dots of light. And by stimulating a pattern, it should be possible to convey a rudimentary form of vision. So this is what such a system might look like. So this is a camera, then a pocket processor that translates camera images into brain stimulation patterns that need to be sent wirelessly to the brain. There's a brain chip and then some electrodes. So we implanted 1,000 something electrodes. This is the work of Xing Chen. Okay, this is the connector, and these are the 16 Utah arrays. This is how we implanted them in the visual cortex. This is primary visual cortex. There were 2 arrays in area V4. This shows you the receptive fields, also in the second monkey. So this is basically where we can produce those phosphines, these dots of light. Here we're stimulating only one electrode at a time. And the purple dot is where the animal directs his gaze. It starts the trial by looking at the fixation point, and when we then stimulate one of these receptive fields, so to say, then you see the animal makes an eye movement there. That had been known, and maybe we made a record by doing this for hundreds of electrodes. It was not so surprising. I'll go a bit quick here. So one thing is that if you look at the current, the minimum current for a particular electrode before the animal perceives the phosphine, that varies across electrodes. Some electrodes might have 10 microamps, other electrodes may have 80 microamps or so. And if you have 1,000 electrodes, then you would have to set a threshold as a user of the system for 1,000 electrodes, and every electrode takes maybe 10 or 20 seconds. That's just a lot of time, so you don't want to do that. So the good news is that we can actually read out the efficacy of V1 stimulation by recording in V4. So here you see that this, for this electrode, the animal did not report a phosphine for 10 and 9, for 7 and 9 microamps. You see there's also hardly any V4 activity, but if you start to elicit activity in V4, that's a good predictor of whether the animal is going to perceive it as a dot of light or not. And that was generally true across many of our stimulation experiments. Now, I think the most important finding in this study was that we can also produce pattern perception. So we trained the animals to recognize letters. So here, for instance, the animals were not blind, so they could perceive the letter D. And then there was a choice menu, and the animal was supposed to make an eye movement to the letter they just saw. And in the most exciting part of the experiment, we did not present this letter anymore, but just stimulated a number of electrodes whose receptive fields we knew, and we knew that they would kind of outline a particular letter, here the letter A, hoping that the animal would make an eye movement to the letter A. And this is basically my last slide, my last data slide, if I can get it to work. So here you see our microstimulation experiment. We stimulate in the shape of a particular letter, letter A here, and the animal indeed makes a corresponding eye movement, picking the right letter from the choice menu. So I can tell you, when we saw this result, we made a small dance in the lab. And, oh yeah, one semi-last slide. Sorry about that, Walter. So we also did this in humans. Actually, this is mainly done by Eduardo Fernandez and his team in Spain, in Elche. So there they implanted only a single UT array in a human patient who had been blind for more than 15 years. And the good news is, when they stimulated multiple electrodes in the shape of a large square, the subject saw a large letter O. Here she saw a small letter O, so that looks like kind of predictable. Here she saw a letter I, maybe, and here she saw a letter L. I don't see it. So it's not so predictable, maybe, but I think what's going on here is these electrodes are all very nearby, so you could easily produce some interference patterns if you stimulate electrodes that are relatively nearby in visual cortex. Now, there's still some work to be done, because this shows you the phosphine locations in the patient Berna. She's actually a co-author on the paper. And so they're very close to the fovea. This is the visual field. It's 90 degrees and maybe 70, 80 degrees in the vertical direction. And this is where she can see dots of light, right? So you have to realize that primary visual cortex in humans is 25 square centimeters on the left, 25 square centimeters on the right. And here we are only addressing 4 by 4 millimeters. Okay, so there's still a lot of work to be done. Okay? So I will skip the conclusions because I already gave them. But I do want to mention the people who did the work. So recordings were made by these people. Figure ground, particularly important role for Lisa. The concept cells, important role for Doris and Judith. And this is the visual prosthesis team. Thank you very much.